Good day, Homo sapiens. Welcome back to studying the Koch microbiology. And yesterday we studied the golden age of microbiology and just some Koch postulates. And today we are moving further in the discoveries by the scientists. And I know it will be taking long. Wait, was just checking if the live was working. And after checking it, let's go back. So what I was saying that we studied the golden age of microbiology yesterday and the Koch postulate. I'll just revise the scientists since we are studying the scientists and discoveries, so you don't forget the previous scientist before moving to the future scientists. So in the golden age of microbiology, like previously they discovered the bacteria and the fungi, etc., and specifically in the case of spontaneous generations. But after that they were recognizing the relationship between the microorganisms and disease. Like for example, Agostino Bassi was the first one to say that the microorganism could cause disease in a silkworm. Like the first discovery about the bacteria or the microorganism, it was actually a fungi. The first microorganism that was discovered in relation to the disease was the fungi, and that was the silkworm disease. By the way, the diseases or disorders or physiological changes caused by the microorganisms are not usually called diseases. They actually are called infections. So keep that in mind. After that, the M.J. Berkeley proved that the potato blight was also caused by the water mold, and this water mold is a protist. It's not fungi, it's not bacteria. Then the Heinrich D. Berry showed that the smut and rust fungi can cause infection in crop diseases as well. So this was about that. With just Joseph Lister, he didn't quite actually discover any microorganisms. He discovered the antiseptic therapy surgery to make the people aware of the methods that we can use to prevent these microorganisms from harming us or from infecting us. And he was impressed with the Pasteur studies. And the Pasteur's era was over, and now we had the Koch's era. Koch is a pretty big scientist in the microbiology field, and he actually explained the method in which the diseases are spread, or how the microorganisms cause infection. So first he was studying it for his own purpose, like on Bacillus anthracis, right, which causes the anthrax disease. But after he studied this, he documented and he demonstrated the postulates which he made on another bacterium, that is the Mycobacterium tuberculosis, and which causes the tuberculosis. He made the 4 postulates in which he showed how the infection works, in which he cultured the microorganism. Then he took the microorganism from the unhealthy animal organism, and then he cultured it. Then he injected it into another healthy organism, and then he growed that microorganism into the culture media as well. The only problem he had with doing this, and when I'm talking about the microorganism and the organism in case of Koch, he did that with the Mycobacterium tuberculosis as in relation to the microorganism, and with the disease, like here it is the tuberculosis. And tuberculosis causes human lung infection, right? But he didn't put the microorganism into the healthy human being. Instead he put them into the pigs, since why you want to let the human, you don't want to let the human die. So when he injected that into the pigs, pig couldn't take it, their body couldn't take it, and they died. And then when he cultured those microorganisms, they look exactly the same microorganism which he cultured from the unhealthy human being. So this was it. Now, the only problem he had was the culture media. Like, he didn't know the exact mechanism or in the exact nutrients that could grow the bacteria, Mycobacterium tuberculosis. Like, if even if he knew that the glucose is needed for the bacteria to grow, he didn't know which type of glucose or source which he should take to grow them. If he went with the solid one, then the temperature wasn't working, and if he went with the liquid one, so it wasn't that sustainable. So his co-worker, as shown in this diagram, where is it here, he was his assistant, and this was his wife. And since they were working with something immunology related, so she suggested that they should use the jelly, which he was working, with her on something research or things. So then they took the agar, right, the agar medium. So but they didn't still know the container for it, like what container would be best to like solidify this agar into and grow bacteria. And then here comes the Richard Petri, which demonstrated, like, which he told that, yeah, Petri dish could be used, and it was named after the scientist Richard. So this was just the summary. If you guys think I shouldn't be summarizing the previous lecture, you can tell me, I won't be summarizing it if you think it's a waste of time. So we are going to move further from this, right? Like, it is going towards the immunology, and we are moving from 1800s to and moving forward to the 1900s. But I think before that there are these kind of things, like the disease, Koch's molecular postulates. All right. So we studied just the postulates, right? What are the molecular postulates? We don't know about this. Why did it go there? Did I said something about moving it to another page? All right. So even before moving to the Koch's molecular postulates, we should, I think, study this diagram. Or should we leave this diagram and study the Koch's postulates, since we are already discussing the Koch? Yeah, we should study the Koch's postulates, molecular postulates. I'm sure a lot of people wouldn't know what the molecular postulates is. Neither do I know. I might have some idea, but didn't know the exact points. So let's start reading. Although the criteria that the Koch developed for proving the causal relationship between microorganisms and a specific disease have been of great importance in medical microbiology. Yeah, they are saying that Koch's postulates were great, importance in the medical microbiology, but it is not always possible to apply them in studying human diseases. Yes, human diseases are complex. Like I said, he had some problems coming up with them. Like, here it is. For example, some pathogens cannot be grown in the pure culture outside the host. Yeah, somewhere, what do you call it in the microbiology word? Facultative? No, not facultative. Intracellular, what do you call it? Viruses are what? Facultative. Oh my gosh, I forgot the name. Wait. We have to know it. I can't. Obligated. Yeah, I don't even need to search it. Obligated. These are obligated parasites or obligated intracellular parasites, or microorganisms. They can't live without the cell. Like in case of bacteria, it can survive in water, it can survive in food, but some bacteria cannot grow outside the cell. And not just the bacteria, like viruses are completely obligate. Like, they can't even reproduce. Well, they can survive when they make themselves crystal, but they can't do the infection and the reproduction part. So for example, some pathogens cannot be grown in a pure culture. Guys, by pure culture I mean when you grow the bacteria, there is no other contamination, no contamination of any other bacteria. It's just the pure culture of the pure organism, the specific organisms you are growing. Because other pathogens grow only in humans, their study would require experimentation on people. Yeah, about the bacteria that can grow only intracellular way, they need to be experimented within their host, and we cannot do if it's the case with the humans. We can't experiment on human if we don't even know the risk and harms, pros and cons of them. The identification, the isolation, cloning of the genes responsible for the pathogen virulence have been made possible. A new molecular form of the Koch postulate that resolves some of these difficulties. So what kind of things like eased the difficulties? First, the identification, like after identifying what can we culture and what we cannot culture. And isolation, yeah, isolation is a difficult thing as well. Like how would you know you are isolating that specific bacteria? Identification can come after that, but first you need to know where to isolate it, how to isolate it. Then the cloning of genes, guys, this is very advanced. The genes cloning isn't, that much in the history. It happened like near to the 2000, not completely near, but not that previous, like not before 1800s, or I think 1900s. Yeah. So cloning of genes responsible for pathogen virulence. Virulence is the property which causes the disease or causes the infection in an organism. We call that like the virulence of the virus or bacteria or whatever. And in case of pathogen, it includes every microorganism. And I don't think it's just microorganism. Like pathogen can be any organism. No, sorry, pathogen can be only microorganism. Wait, helminths are pathogens? No, I think pathogens are specific for microorganisms. Virulence have made possible. So there are genes that encode the virulence factors. Like virulence can be due to some specific antigens, and in case of antigens that attach to the cells of the human cells, like that is actually the virulence factor. If they didn't have that virulence factor, if they didn't have that receptor, if they didn't have that identity protein, it wouldn't be possible for that microorganism to attack the human being, and it hadn't been possible for them to become pathogen. So that's why we clone the gene responsible for virulence factors, since we all know that the receptors, proteins, or these kind of things are made by the genes or are coded by the genes. So identification, isolation, and cloning of the genes made it possible for the easing of the difficulties. A new molecular form of Koch postulates. So this became the molecular form of the Koch postulate. How did it become? We are going to study in these steps. The emphasis is on the virulence genes. Okay, the emphasis part is on the virulence gene present in the infectious agents rather on the agent itself. Okay, so about the agent, they are talking about the one that is being attacked, and the infectious agent is the one that is doing the attacking. So they are saying we study the virulence genes of the infectious agent, right? Not the agent is not the one that is being attached. Agent is the complete organism, right? But there is something on that agent which we call the infectious agent. Like for example, this is a bacteria cell, and there is a receptor on it. So the bacteria is antigen and the receptor is the infectious agent. So we only study the code for that infectious agent when we are cloning the gene, since we have gene for everything and we cannot study the whole gene. The molecular postulates can briefly be summarized as follow. First, the virulence trait under the study. If we are not searching anything, I think I should make this smaller. Okay, so the virulence trait under the study should be associated much more with the pathogenic strains of the species than with non-pathogenic strains. Okay, so they are saying that the virulence trait that we are studying should be associated with the pathogenic strain than the non-pathogenic. It's common sense. If they are non-pathogenic, if they cannot cause disease, they wouldn't have that pathogenic trait or virulence trait. So how are we going to study that? So we can't even study it. It's not even a possibility for us to study the virulence factor. So the virulence factor that we study should be associated with the pathogenic strains. And by this strain, it doesn't mean species. in the species and in the subspecies there are organisms that are only different from one another by just one characteristic. By one characteristic I mean just single characteristics, like it may be a single polypeptide in their enzyme or in their proteins, like this much difference, and that's how they become strains. And so that's why it's just a slightly bit coding in the gene that differentiate them from one another. So we shouldn't be studying the unpathogenic strains, we should be studying the pathogenic strains. The second is inactivation of the gene or genes associated with the suspected virulence trait should subsequently what am I saying? Inactivation of the gene or genes associated with the suspected virulence trait should substantially decrease pathogenicity. All right. If they are saying that if we deactivate their virulence factor, it should also decrease their pathogenicity as well. Like, right, if the sole way of the pathogen or the bacteria to infect another cell is due to their only virulence factor, then inactivation of the gene will do it. Inactivation of the gene will work. But if there are some that much, usually the virulence factors are coded in the gene, but there are cases in which they are not coded by the gene, like it's something. Inactivation of the gene or genes associated with the virulence factor should decrease the pathogenicity. Usually it should happen. It should happen. There is no way if we inactivate gene and the pathogenicity still remains. That's not even possible. But they are still saying substantially decrease. They are not saying that that inactivation of the gene will completely diminishes their pathogenicity. Then the third, replacement of the mutated gene with the normal wild type gene should fully restore the pathogenicity. All right. They are saying if a microorganism is non-pathogenic, like if it doesn't have its virulence factor, right? And we put a mutated gene that has the virulence factor, that would make, and we put them in that non-pathogenic microorganism. So of course that will make it pathogenic. This is one case. But in this case they are saying if we inactivate the gene and they loses their virulence factor, and if they come in contact with their original type of gene, and by some cases like conjugation or environmental transformation, they pick up their wild type gene, like their original gene that was inactivated by us humans, and if they pick that wild type of gene, it will restore their pathogenicity, like its dominant culture. Like even if their original gene is non-mutated, but the wild type gene enters into them, that inactivation isn't going to work. They will remember that yes, this is their gene and this is the original form of their gene and this is the original work of them, like they should cause the virulence and they should have the virulence factor. So it will just replace the work that we have done it. And then the gene should be expressed at some point during the infection and the disease process. Now, normally the gene should be expressed at some point during the infection and the disease process. Like if the replacement occurred and if the pathogenicity is restored, then this gene is going to get expressed, this normal wild type gene that they took. Even if the previous one is inactive, this one is going to work just fine. And that's going to be expressed at some point during the infection or the disease. Antibodies or immune system cells directed against the gene product should protect the host. Antibodies and immune system cells directed against the gene products should protect the host. Okay, so they are saying if our body antibody and immune system cells attack that virulence factor. By the gene products, it just means the virulence factor. Like the virulence factor can be toxin, the virulence factor can be antigen, the virulence factor can be what else could it be? I don't know, penetration, something like this. So they are saying if our antibodies and immune system cells attack that virulence factor, if even if they don't attack the gene itself, even if they don't harm the gene, if only they attack the gene products like some antigens or some toxins, it should protect the host. And that's usually what our immune system do when they fight against the microorganisms. They bring the antibodies and they attack, like they attaches to the receptors, and they become inactive, or the cells just engulf the whole cell, and it protects the host as well. So that was the molecular approach. The molecular approach cannot always be applied because of the problems such as lack of appropriate animal system. Okay, so they are saying that it cannot be applied because of the lack of appropriate animal system. Like, it's not your usual routine to deactivate your genes and then the wild type genes again pick up the pathogenic, which was rendered unpathogenic, pick up the wild gene and again regains their pathogenicity. It is not applied because of the lack of the appropriate animal system. Like, we don't have animals, or not animal system means they don't have a body to experiment it on or something like this. It also is difficult to employ the molecular postulates when the pathogen is not well characterized genetically. All right, this point where they are saying if we don't know the genes of the pathogens, like we don't know, even if we know the sequence of the pathogen genes, the DNA, but we still don't know what genes do what, then we can't know how the molecular postulates will work, since we do not know what genes are for the virulence factors, or all the genes that we studied are all related to the virulence factor, or are there more that we are leaving in case of the virulence factor. So, by the way, I don't think this will work. This book is the 7th edition, so it's quite, it's, I think, updated till 2005 or something. These days we have quite grasp on the genetic code or the sequences, and they are working in a lot of microorganisms. So let's revise what we studied under the Koch molecular postulates. First, we should study the virulence traits of the pathogenic strains. Then we should, if we inactivate the genes that are associated with the virulence trait, it should decrease the pathogenicity. But if they pick up the wild type gene, the replacement of the mutated gene will occur. Like, the one that we inactivated will be replaced with the wild type gene, and it will regain its pathogenicity. Then that gene should be expressed, like which is replaced, should be expressed at some point in the infection. And if our antibodies and immune cells attack these virulence genes produced by products, like if they are attacked, it should protect the host. So this was it, and we should go back to the what this table or what this section is supposed to be. The techniques and applications. Scientific method, techniques and application. I don't know what they are implying here. Although, are they saying that how the scientists work step by step? Let's study this flow chart before going to the wordings. So first we have problem. We always start with a problem in research. First we have problem, then we develop a hypothesis. Like, this problem is this, and it may be affected by this, or this is the result of this. Like, different kinds of hypothesis. By the way, what is hypothesis? I'm just wondering about its original meaning. A supposition or proposed explanation. Okay, it's just a supposition here. Supposition or proposed explanation made of the basic way here on the basis of the limited evidence as further investigation. Okay. So this is the proposition which is done in a limited conditions. Select the information needed to test the hypothesis. Now that, for example, let's deduce a problem from our cell. Like there is a tuberculosis. Wait, no. Like there is a malaria. Then we develop a hypothesis that mosquitoes are the cause of hypothesis, right? I know this is wrong because normally the malaria is caused by the plasmodium. Yeah, Plasmodium falciparum or vivax or these kind of plasmodiums. But we are saying that the mosquito causes the malaria. Then select the information needed to test the hypothesis. What kind of information? Like for example, in this case, we will collect the mosquitoes. Then we will see the environment of the patient, like if it is clean or not, or if it happens in the dark or in the day, like this kind of information. Then we are going to test it. Collect the information by observation or experiment. Okay, like in this case we are not doing an experiment since it involves the humans. In this case we are doing observation, and in case of Mendel's that was an experiment in which he was watching the change of the genes, change of the characteristics due to the genes that he mutated. So it can be an experiment and it can be observation. Then we analyze the information. Okay, now we have information which we collected from the environment. Now we analyze it. Like, if is our hypothesis correct according to the information that we gathered? We still don't know how it correlates, but now we are going to correlate. Then the hypothesis not supported, hypothesis supported. Like in the case of Mendel's, in which he studied the characteristics were changing due to the gene change, that hypothesis was supported. But in the case of Plasmodium, no, mosquito causing the malaria was not completely supported. Like it's halfway. Like of course malaria is the transmission channel, so it is kind of source of malaria, but again it's the specific type of mosquito that would cause malaria, so it's not completely true. And in science we need the perfect answer, we don't need half-baked answers. So it's not supported. Exposed to more tests. In case your hypothesis is correct, now we know we are going in the right way, so that's why we are going to expose it to the more tests. Then what happened? Hypothesis or theory not supported. Okay, originally you thought yeah this is working just fine, or wait, in this case, like I said, it wasn't completely supported, it was halfway supported, so we put it in the hypothesis supported category, and after that we exposed them to the more test. But hypothesis or theory not supported or continued support. Like in Mendel's case, it was completely supported. Like that's why it was even if you tested it multiple times, the answer remained the same. But in case of mosquito, I think it didn't remain the same. Like first, even if you expose someone to the mosquitoes, there is no way that every mosquito will cause the malaria. So the experiment wasn't completely success. There must be some healthy organisms left that weren't harmed by the Plasmodium, or you can say mosquitoes. So it wasn't supported, so they left it. Then theory, then more test, then continued support. Like after more test, it doesn't matter at what stage it can't be supported. Like in malaria, I think it would made more clear that maybe the mosquitoes weren't the cause, but in Mendel's case it was continuously supported because the tests were not failing. And after multiple tests, and if it doesn't fail, it becomes the law. Mendel's law, if you remember. There is no malaria law. Alright. So now we are going to read it. What this book is trying to say by this flowchart. Although biologists employ a variety of approaches in conducting research, microbiologists and other experimentally oriented biologists often use the general approach known as the scientific method. Alright, they are saying that the general biologist, disrespect to them, general biologists. Alright, so the biologists are, the one that don't need like that much testing or things, like they are clear about, or they don't have any problem to begin with, right? Like if they are just studying the plant, generally plant, don't have problems, so they can approach multiple ways to handle their experiment or their research. But they are saying microbiologists and other experimentally oriented, like it has to be experimental. Biologists often use the general approach known as the scientific methods. So this happens in experiments because in experiment we have controlled environment and we can do the tests multiple times. But in case of wild studies, we cannot do the experiments multiple times. That's why maybe they acquire different types of approaches, and in our case we acquire the scientific method, since we can repeat it over and over again with the same type of microorganism, with the same type of conditions. They first gather the observation of the process to be studied, then develop a tentative hypothesis. I think tentative means what do you call it? Like that is not perfect. Like it's just hanging somehow, like not solid, or maybe practical, like putting it into words. Not certain or fixed. Yeah, done without confidence. Like you are just stating whatever you come to mind when you see it somehow. So that's a tentative hypothesis, an educated guess. Like you are guessing, but it's not baseless. Like you have seen something and then you are going to say like it's happening this. Like in case of malaria, you have seen the people are that have mosquitoes around are getting infected. That's why you said that mosquitoes cause the malaria, but that is educated guess. Like you are not saying that animals cause the malaria, right? Like you haven't seen the animals around the patients. Like there may be, but it's not to all patients. Like that won't be an educated guess. So by educated guess means that you are making a hypothesis related to the environment you are seeing to explain the observation. Yeah, you are just explaining it, that yeah, look, these are because of mosquitoes that the malaria is being caused, even though it might not be true. You are just saying it's because you observed these kind of things. This step often is inductive and creative because there is no detailed automatic technique for generating hypotheses. Yeah, there is no technique or anything, it's just your guess. Next, they decide what information is required to test the hypothesis and collect this information through observation or carefully designed experiment. We already explained it. After the information has been collected, they decide whether the hypothesis can be supported or falsified. Yeah, after the information has been collected and then they compared with their hypothesis, then they already know that if it is correct or if it is not. If it has failed to pass the test, the hypothesis is rejected and new explanation or new hypothesis is constructed. If the hypothesis passes the test, it is subjected to more severe testing. Like they are first testing in a generalized way, then they are going to specific and narrowing their testing methods until they reach a point where they can no longer narrow the test anymore. Alternative hypothesis, and then refining the hypothesis that survives the testing. Yeah, after the hypothesis survives, they refine it more, like they narrow it more. This general approach is often called hypothetical deductive. Hypothetical deductive method. Well, deduction is deduction and hypo is hypo. Did I make sense? Hypothetic is like it came out of your mind, like the idea, and deduction like you made a result or an outcome based on the experiment you have done. So one deduces prediction from the currently accepted hypothesis and tests them. In deduction, the conclusion about the specific case follow logically from a general premise, if then reasoning. follows logically from general premise. All right. Like, how are you going to conclude? Like, there is a rule for you and then the result follows. Like, if this happened, then this will happen. If this didn't happen, that won't happen. Like, if this didn't happen, that will happen. Things like that, reasoning like that. Induced deduction is the opposite. No, induction is the opposite. Okay, one is induction and this one is deduction. And the process that we done is the hypothetical deductive method. So they are explaining what is deduction and what is induction. Alright, so in deduction, the reasoning follows. While induction, I don't think the reason is following. A general conclusion is reached after considering many specific examples. Both types of reasoning are used by scientists. When carrying out an experiment, it is essential to use a control group as well as an experimental group. The control group is treated precisely the same as experimental group, except the experimental manipulation is not performed on it. In this way, one can be sure that any changes in the experimental group are due to the experimental manipulation rather than to some other factors not taken into an account. Wait, what do they mean by the control group and the experiment? Oh, I get it. The control group is which you are sure that if you do this to them, they are going to give this result. Like, it's, how do I explain it to you guys? Like, if you paint a blue paint on something, it's going to appear blue. That's set in stone. So that is your control hypothesis, right? But if you have an experimental group and you don't know what color is this, right? And you put them on the paper and on the wall, and if it looks blue, that certainly means you put the blue paint, right? Like, that is your experiment, because you know that you don't have information about what you are putting. But if the result that comes the same as control group, then you automatically knows the step before that. But if your result didn't come blue and it come yellow for some reason, then you know that the step, that the one thing that you put on your brush isn't the blue color, it is something else. That means that your experimental group is not the same as control group. Your experimental group should give the same answer as the control group, or how it betrays it. Wait. Like, this is usually done, I am not sure about the general process, this is usually done in the antibiotic resistance, or checking the positive negative test in case of this. Like, alright, let me explain them in the words which I understand. You take blood, which is, you don't know if it is infected with malaria or not. Now you have certain kit which has the malarial antigen and which has the antibodies, right, against it. You put the blood that has malaria in it and that antigen which you already have, that antibody you already have, when you put them together, they form complex. So this is your control group, that you know that the malarial antigen will react to the antibody and it will make a complex, right? This is your control group and that you know it. Now you have antibodies, right, that you have prepared, but then you have a blood and you are checking it. Alright, the control group was positive or negative? It was positive because, the blood had the malaria, that's why it made the complex. That's your control group. Then you have the experimental one. You have antibodies for anti-malaria, right, for malaria, against the malaria. Then you have blood. Now you don't know if it would have the antigens of malaria, like if it has the microorganisms that are causing the malaria or not. So how will you know if it has it or not? When you put it against the antibodies and it don't form complexes, like it doesn't react the same way as your control group, that means it is giving the different reaction, and there is only one possibility of that, and that is you don't have malaria, means you come out negative. But if it reacts the same way as control group and makes the complexes, that means you are positive. and the same as control group. So control group is a set in stone procedure or set in stone process that you know through and through what made the result come the way that it came in the control group. But in experimental group or in experiment, you have one thing that you know and one thing you are experimenting on to deduce what will come, what result will it come. All right. Still, I will search it in Google for better explanation. What is, I hope I am not saying it wrong. Group of subjects in an experiment that do not receive the treatment of being studied. Yeah, we are not studying it. We already know the result. It is used as a baseline to compare the result of the experimental group. Yeah. Thank goodness I wasn't wrong. I blabbered so much and I thought if I was going to be wrong, that would ruin my mental health. It's a group activity that allows people to learn about the group dynamics and the processes through personal experiences. All right, they are talking in the world of people, social, like psychology. We are not talking about this. In our cases, they are not people. In our case, they are microorganisms. All right, so we now know that. If a hypothesis okay, where were we? We went way too deep into the control group and in the experimental group. So what we were studying that, yeah, so the result of the hypothesis can be hypothetical, deductive, or can be inductive, right? When carrying out an experiment, it is essential to use a control group as well as experimental group. Yeah, it is essential. So you better remember when you are doing research or internship or whatever. If a hypothesis continues to survive testing, it may be accepted as a valid theory. First, it becomes a theory. A theory is a set of propositions and concepts that provides a reliable, systematic, and rigorous amount of an aspect of nature. That means that it is passed through a lot of testing and the systematic testing. Systematic testing means it is went through hurdles step by step. Like we know what comes first, what comes after, and it's reliable because we know the procedure and we have done it several times, and you can do it too if you wanna see the result the same way. The result will come the same as done in the lab. Rigorous means, yeah, it's a hard work because like how many times did we do the same test or not the same test for just a single hypothesis? So it is important to note that the hypotheses and theories are never absolutely proven. Scientists simply gain more and more confidence in their accuracy as they continue to survive testing, fit with the new observation and experiment, and satisfactorily explain the observed phenomena. Like they are saying that is not set in stone, but till now what they did, the experiment and the result didn't betray what they were doing, and the result will come the same way again and again, and it will survive testing. That's why they, called it theory, because nothing was changing. But then what? Ultimately, if a support for the hypothesis or theory becomes very strong, like it can no longer be denied that the result is going to come the same way, whatever you do, whatever way you go across it, that's when it becomes a law. It is considered to be scientific law. For example, examples include the laws of thermodynamics. All right, thermodynamics is like temperature always, make themselves stable or balanced. Like if you have two bodies, hot body and the cold body, when you place them close together, the temperature is going to be the same for both of them after some time. By the way, the temperature go from the cold body to hot body to cold, or cold body to hot. Temperature go from, wait, oh, when did this happen? go from hot to cold or cold to hot. This is also set in stone. Heat flows from hot to cold objects. Right, of course the heat is in the hot objects, not in the cold objects. That's why it goes from hot to cold. And let's see what is the law of thermodynamics. I hope I'm not wrong. That define a group of quantities such as temperature, specific laws. Okay, so there isn't one law, there are 3 laws. And I was talking about one law, like here. Wait, isn't there a clear diagram of it? First law of thermodynamics is the heat transfer. Or wait, conservation of energy. Yeah, whatever you do, the energy is going to remain the same if you finalize or you add the final and the initial. Then the second law, yeah, the heat transfer. Then the third law, entropy. Yeah, that the world is going towards the disorder. Normally we think we are going towards the order, but if you look at the world, things are going in the other way. If you look at the tree, the leaf falls and it goes more and more towards the entropy. And even if you look at the world day by day, the things are not, recycling itself or saving itself. It is going towards more destructive sides. So this was the scientific method. Okay, so they are saying how the scientists do the research. This was all about that. So now the viral pathogens. Should we study more? I don't think we should. I think we are done with today. The Koch molecular postulates and the scientific method should change since there is no scientist in it, it was just the general knowledge. Except this Koch, and we just don't know what is there to study more. There is the immunology, the development of wait. What? Where did the Google go? Or wait. Wait. Something happened, which I don't know what happened. Okay, let me shorten this so we can see. Alright, the immunology studies are remaining, the industrial microbiology and ecology. Okay, after that we are going towards the scope. But it still isn't the same as scope because they are talking about the 1929 and this and that. So this is not in order. And then the future of microbiology. I don't know why I'm reading the 7th edition, but it doesn't have, that much future in it. The future that they will be stating is already done. But at least we can prove it if it's already done in our age. Or if it still remains to be done after 2025 or in 2025. And then finally we are done with the first chapter. How many days will it take? Wait, if we study one page at a time, 1, 2, 3, 4, 5, Thank goodness. Alright, this is it, and I'm ending this stream.